College UK Cambridge Centre podcast. In this Integrated Cancer Medicine Research in Focus series, I talk to various ICM members about their research and how it is supported by the vision of the Mark Foundation Institute for Integrated Cancer Medicine. MFICM research uses cutting-edge analytics to maximise the use of diverse high-volume datasets and by capturing cancer heterogeneity in time and space in patients receiving active treatment. Integrated cancer medicine aims to transform the way the world treats cancer by affecting patients along their treatment pathway and ultimately accelerate cures.
Today I have with me Dr Dario Bressan to talk about the IMAXT project and how it fits in with the Integrated Cancer Medicine vision. Dr Bressan is head of the IMAXT laboratory at the CRUK Cambridge Institute at the University of Cambridge. So thank you for joining me today, Dario. Can I ask you to start with, could you please outline what the IMAXT project is?
Yeah, so gladly. To begin with, the name, it stands for Imaging and Molecular Annotation of Xenografts and Tumors, which is a mouthful, I think we can all agree. In the beginning, it was just IMAX, because we wanted to capture the idea of doing things in large resolution and in space. So, of course, IMAX Cinema. We had to put a ticket that they would sue us otherwise, and they'd probably win. But the key idea is making a three-dimensional single-cell resolution map of a tumor. If you think about Google Maps. Now, Google Maps, in a way, have changed things for looking at maps, from just looking at a paper, something to look at an interactive map where you can zoom in, zoom out, click on individual buildings and know exactly what's inside, how they are connected, how you go from A to B, and so on. That's an infinitely easier problem than what we're trying to do. First of all, because, the surface of the Earth is two-dimensional and the tumor is three-dimensional. And second, because the number of information that you need to capture to really know what each element of the tumor is doing is infinitely high. So we are stringing together a series of technologies, all of them pretty cutting edge, some of which didn't even exist when we started, to build this interactive map that you can literally enter inside. And I'm not actually joking. We have a department that does virtual reality when you have an app that you can literally enter inside of the tumor and analyze it. And the idea is that having all this information at your fingertips, the whole molecular makeup of every cell of the tumor at your fingertips, give you a much bigger chance to understand how the tumor is evolving, how it's responding to therapy, how it's changing in order to avoid the intervention, how it's proliferating and propagating outside of the main tumor to form metastasis. And ultimately, this all goes back to better diagnosis, better stratification of therapies that we already have, to target the right patients and potentially development of new therapies altogether.
And you can actually walk inside the tumour with a VR headset on, is that correct?
That's correct, yes, it's pretty cool.
That's very amazing. Are you able to take different shots of the tumour as it develops and as it responds so that you can see the developing, or is it just a one shot of this particular tumour?
So the technologies that we have are disruptive, meaning that the sample that we are imaging is done, doesn't exist anymore afterwards. We can take a shortcut to that by looking at biopsies, for instance. So we can look at subsequent biopsies of the same patient, or if we use animal models, which of course it's always a plus and minus situation, but if you use animal models which are relatively stereotypical, where a tumor tends to develop always in the same way, you can take it at different stages, or before and after therapy, or with and without, and then make a comparison. Actually, that's a pretty large portion of what we do. So yes, it doesn't give you the dimension of time immediately out of the box, but there's ways to trick yourself into it.
Amazing. And so when did the project start and how is it funded?
The vision for this started actually in 2014, 2015, where our laboratories, I'm part of the Hannon laboratory before we sort of branched off in what we are now. We moved here from the US. We used to be close to New York. And I've always had this dream of doing spatial measurements, of doing sequencing, but being able to do it in space so that you would preserve all the relationship between cells. And the time being, the laboratory was working on breast cancer already, did a pretty long history of doing that. I was working on a number of projects holding this idea of spatial profiling. The catalyst for really all this to happen was then, I think it was 2015, Cancer Research UK came up with the Grand Challenge Initiative, which was a pretty blue sky idea, right? It was, we're going to put £20 million on the table for multidisciplinary, multi-laboratory groups to fix the biggest challenges in oncology. And here's a list of challenges, which all of those essentially now are impossible. The idea is that we're going to give you a ton of money and 5-6 years to fix it. One of them was, can we map a tumor cell by cell? And I think Greg and I had a couple of email exchange back and forth and said, look, this is us. We're going to try this. And then we involved most of the rest of the laboratory. We actually had some close collaborators that were already very interested. We branched off to reach some other collaborators. And so, fast forward, we applied for this, and we actually got one of the first 4 grant challenge grants to do this. And then with that incredible amount of resources, we started in 2017, and we've been going until now. And we are now in our last year of the grant, where we're starting to reap the fruits of what we saw at the beginning. And I want to stress that it was a very, if you want, dangerous and ambitious thing to do for a funder, because we haven't actually got a lot for the first 2 or 3 years of the grant, because there was so much infrastructure to build just to get people to collaborate, just to have the computational system to handle this data. Nothing existed, right? That was science fiction. And then before you started to get anything out of it, there was a very long lead time. But they stuck with us, and we stuck with ourselves. So now we finally have something to show for it.
Yeah, it's an amazing project. It's, as you say, an amazing thing to have the vision to fund, an amazing thing to have the vision to build. So would this technique work with other types of cancer? You've mentioned it's specific to breast, but would it work with other types of cancer?
Well, absolutely. As a matter of fact, we are branching off to other types of cancer now. We needed to start from somewhere. We started from breast for 2 or 3 reasons. One of it is that it's clearly, especially triple-negative breast cancer, it's a cancer where the need is enormous. If you think about it, 1 in 7 women worldwide will get it at some point, right? And at least for some of them, prognosis is pretty good. For some of them, there's still a lot of problems, and some of those can come back 20 years after they've been treated. So there was clearly a lot of need. The second is that it was the kind of situation where we know a lot about it. There's been a lot of work done on it, actually a lot of it from this institute, which showed all the genomic diversity and all the information. So we know quite a lot of how it works at the genomic level. I'm sure that you probably have covered before the Personalized Breast Cancer Genome Initiative, where now every woman that comes here gets their cancer sequenced. But at the same point, we knew so little about it, because there was a situation where 100 women come in, they have all the exact same genetic fingerprint of the tumor. 60 of them, the therapy works as you expect. 20, it works very badly. 20, it works much better than you expect. You have absolutely no idea why. And the idea was that it had to somehow depend on all this layer of interconnection of how exactly each tumor interacts with the body that surrounds it, that you could just not resolve with traditional methods. So we thought, okay, we can make the difference here. We can give that step. But the technology itself is agnostic to which tissue you apply it to. So it will work with other tumors. We are applying it to ovarian right now. There's talks of applying to lungs. We've got something looking at renal. So I would say every solid tumor can really work with this. Liquid tumors like lymphomas or so on, less so, because they don't have such a clear spatial structure that spatial methods don't really work. Like any solid tumor should work.
You've touched on it a little bit, but what data does the project incorporate?
There's no single technology that can give us all that we're looking for. So ideally, you would want a box where you put a piece of a tumor in, you get out with a complete 3D model with all the information. In reality, it's not like that. Some technologies are really good at giving you gene expression information. Some technologies give you protein expression information, but they don't do it in 3D. Some technologies can give you 3D images, but they don't give you much information at all. They basically just give you images. So what we do is we start with the tumor. We embed it into a block of a very hard gel, some sort of matrix. Then we put it into an instrument which is called serial photon tomographer. It's essentially a giant room-sized microscope, which is kind of the love child of a microscope and a microtome. So it takes images of the tissue, but then it also sections it in very, very paper-thin sections, which then get automatically collected. We have a robotic arm that picks them up and collects them on slides. That gives you the two-dimensional structure of the whole thing. Then these sections get taken and get routed through two other technologies, which are called MERFISH and imaging mass cytometry, plus a few others as well. And those either give us gene expression or protein expression. A very high resolution, very high multiplicity, so we get all this molecular information. And then we have a lot of computational tools which were developed between us and the Institute of Astronomy. Actually, we needed astronomers to do this because the data size is so large that they knew how to handle them. We did not. All this gets put together into a coherent, integrated model. And then sort of on the edge, as a periphery to this, we have more traditional method, right? Regular sequencing, single-cell sequencing. It's kind of amazing that I refer to single-cell sequencing as a traditional method where it's kind of less than 10 years old and it's already considered very cutting edge, but it's almost like what we started from. And so it's a lot of data that then gets put together. Just to give you an idea of the size, the sort of compiled information about a tumor is something like 10 terabytes or something like that. So it's like a couple hundred years of non-stop music.
That's great that you defined that. And amazing the multidisciplinary side of you joining up with astronomy to make that work.
Well, that's a nice story, right? Because we're going to stream lots of samples. Each one's going to be a few terabytes of data. And our IT department was like nervous about it. And astronomers were like, oh yeah, that's Wednesday. Well, I mean, the group of people in the Cambridge Institute of Astronomy that we work with, their project, which is called Gaia, is to map every single star of the galaxy. So that's the scale of data they work with, right? So we said, all right, do you want to just take this and convert it to the galaxy inside, if you want? And they already had very good connections, because actually a lot of the image analysis challenges that you have there are not that different from the image analysis that you have in microscopy. So they were able to very usefully convert this. But that's actually not the only interdisciplinary thing we do. I mean, we work with chemists. So obviously trying to improve technologies, there's quite a bit of material chemistry, but organic chemistry that's involved. And the virtual reality project, we actually work with video game designers.
I was going to ask about that. That's really interesting. And how did you make all these connections? Were they just personal connections, or did you put feelers out? How did you find these experts to work with?
Some of them were already known. So chemistry, we have the Shankar Subramanian, which is one of the best DNA chemists in the world. He's in Cambridge and he's part of our institute as well. So obviously that connection was straightforward to make. The astronomy had already started collaborating with one of the members of our team, Carlos Caldas. So there was a smaller scale but already existing collaboration there, and we just scaled it up a lot. The virtual reality was a lot weirder. That was a random acquaintance of Greg with somebody in CRUK, which had seen a random presentation, and then we wrote an email to this person, Owen Harris, saying, do you want to do this? And he actually thought it was a scam. Sort of deleted it right away. We had to sort of write him again. They said, no, I'm not doing this. I think it's crazy. And then, we basically told him, you know what? We're just going to pay you a ticket to come to Cambridge and just talk with us and see the lab. Worst case, you just get free dinner out of it. And he came and they saw the project and he was hooked in. And then they actually spinned out a small company to work on the virtual reality side of that.
So interesting that it produced the company as well. Amazing collaboration. How does the IMAXT fit in with the ICM vision, would you say?
I think that the IMAXT sort of sits a little bit ahead of everything, if you want, because if you're thinking from a very translational and clinical point of view, the grand challenge is not the project that is going to produce immediate new therapies or immediate new diagnostic tools or insight. It's a basic research and a discovery tool. And I can imagine, we all can imagine, and we hope that some subset of what we do will eventually convert to diagnostics, but that will be sort of in a way the follow-up phase. So the way I see it is that we have put together all this infrastructure to learn a lot more about tumors work and about how tumors evolve. With this knowledge, we're going to take a subset of this infrastructure. So we're not going to get the whole million scale, multi-genomic, multi-transcriptomic profiling, but maybe we're going to identify a subset of, 20, 30 markers that make a signature that very clearly identify tumors that need a different type of therapy. And that's going to go down into diagnostic, that's going to go down into clinics. So I see it as a knowledge generator and as a generator of potential signature and marker panels and ideas that then get followed up and convert into a lot of things that then fit into the integrative medicine aspect. Broadly, I think that the idea is almost edging on personalized medicine. The idea that a lot of tumors, not maybe every tumor, but a lot of tumors are different, and the difference depends a lot on the interplay between a specific patient and a specific tumor. And if you understand how these things work, then you can design a therapy that's much more tailored. The sort of dream that I have is that patient will go into surgery, tumors will be excised, it will go through something that will resemble our pipeline while the patient is recovering from the surgery. By the time the patient is recovered from the surgery, data is back, and the doctor can use it to design the best possible therapy course for that specific patient. That's where I would like to get. It's going to maybe take another 5-6 years to get there, but I think that's the trajectory. And we're already seeing some pretty clear evidence that some of the technologies that we use, like imaging mass cytometry, for instance, there are actually clinical trials already that are using it.
Yeah, so it becomes another data pipeline, really, into the integrated cancer medicine vision.
Yeah, it becomes another data pipeline, and then I might see it as a generator of knowledge. Kind of like if you think that the moonshot, going to the moon generated tens of different technologies that kept being then introduced into normal life on the Earth for like decades after. I think the grand challenge is something going in that direction. Actually, a very good example of it is that there's a new project launched by the Wellcome Trust, which is called LEAP, which is much more translational. They have a very clear aim, which is building a predictor which will guess with 80% accuracy whether a triple-negative breast cancer will or will not recur. And that led in 5 years. So that's a very concrete target with a very big implication of therapy. And we are doing that now. We got the award for doing it. And the technology that fits into it, it's largely technology that we have developed with the Grand Challenge. So actually, CRUK likes to talk about that as a child of the Grand Challenge, because it's almost like, okay, we've got this thing done. We've got this knowledge done. Now we can do all these other cool things that actually are going to have an impact.
What are the challenges of this type of research?
I can answer on different levels. There's the technological challenges. In that is that all these technologies that we use, they were basically at the stage when there were 1 or 2 papers demonstrating them in one laboratory, the laboratory that invented them, on one type of tissue. So that's the level of cutting edge that those technologies were. We had to make them robust. We had to make them work on a different type of tissue, and we had to string them together. That wasn't trivial because the technology that used proteins as a step required destroying everything that was not protein. The technology that was measuring RNA as a step required removing all the proteins. So it wasn't trivial to find a way to connect them all together. The technology that produced the sections wasn't compatible with any of those other 2 things. So there was a lot of technical challenges. It was like, okay, we want to go to the moon. We're kind of at the point of having a hammer and nails and try to put together things. But that, I mean, it was what the job was. It was what we were given the grant to do. And we knew we had the right people to do it. The challenge, the level of logistics and organization, was that we have about 10 laboratories which span something like 6 time zones or 7 time zones. And we had to make this work, right? Some of these people hadn't met together, so we had to make them meet together. Some of these people didn't speak the same language. I mean, they all spoke English, but they didn't speak the same technology language, right? The astronomers had no idea what genomic coverage was or other things. And I had no idea what a shader was in, virtual reality language. So I think there was a lot of time that had to be invested by everybody to learn everybody else's language in a way. And then there was the heroic job of the project manager, which is sort of the unsung heroes of this. People that take care of scheduling the meetings, making sure that people go to the meetings and think about plans and arranging things. And we had the luck. I mean, the first one was Ilaria Falciatori, which is now moved to a startup. Currently, we have Heather Rushmore. They are incredibly good, and they took on a job that was incredibly difficult. I mean, also Erica McKenzie, Sylvia Zul. I mean, we had quite a few people coming around and doing the job. And then all of this sort of eventually ended up solving this giant logistical issue, which was making this thing work.
The project obviously spans many different disciplines and geographical locations. Could you tell me any more about perhaps some of the opportunities and challenges you didn't plan for at the beginning of the project, or something that's come out of the project as a byproduct?
Challenges that I didn't plan for at the beginning of the project. I mean, I don't think anybody was thinking that in such a collaborative project that requires constant meetings, constant interaction, sending samples, 2019 happened.
Oh, yes. That little thing.
So that little thing where, traveling the world, basically ground to zero. People could still go to work, but in different countries, laboratories missed anything between 6 and 9 months, and nobody could talk anymore. We basically didn't have any in-person meetings for 2 years. So we had to adjust, lots of Zooming, lots of finding new ways to collaborate. That was definitely a challenge. Unexpected outputs of this, I would say that some technologies that sounded really, really viable at the beginning ended up to be more problematic, but some other things that we didn't initially consider turned out to be really useful. And some collaborations that we started, they are sort of the gift that keeps giving, right? Even now that the grand challenge is finishing, there are a few of the laboratories where we have these collaborations that have now produced another 2 or 3 grants that we're sort of exploring. And it's really the fact that we have started a number of friendship and really close contacts, which now suddenly make stuff that wasn't possible before possible.
It's taken on a life of its own. Almost.
It's taken a life of its own, and there's no stopping it. I mean, the company made for the virtual reality, they're branching off, they're launching the software, but they're thinking about keep going and essentially producing a viewer for three-dimensional data that, again, it's probably going to take a life of its own. We're doing the ovarian project, the welcome lead that I was talking about. And even from doing all this work and understanding the limitations of the technologies we are working with, we are actually in the position where we are figuring out, okay, what is the next step, right? I told you at the beginning, the dream is a box where you put in a tumor and the stuff comes out. So we are starting to think now how that box can actually look like, because we understand now what are the features that this box has to have inside. So there's a whole legacy of innovation that I think we are starting now on this long end of the grand challenge in a way.
Amazing. Can anyone experience the IMAXT project?
Well, yes, there's a few ways. So we are open for business again, so people can come and visit us. And actually, I mean, I'm not joking, people do that. Cancer Research UK and the CI specifically are very open. So literally anyone can come and say, I'm interested in doing a visit, and we are going to organize a visit. So that's a way if you happen to be, in Cambridge or close by. We are actually part of a museum exhibition. So there is a museum exhibition that was in Birmingham for a while. Now it's at the London Science Museum, which is called Cancer Revolution, which is all about hope and new therapies and what cancer is. And we're actually featured in that. So people can go there's a recording of the virtual reality. There's a description of the project, so people can go see that. The virtual reality itself is about to come up. So we're expecting it to come up around October, which means it's going to be freely downloadable with a few data sets that they've generated in there. And that actually runs on consumer PCs. If anybody who has a virtual reality headset for video games at home can actually download it and look at our data. We're going to both have some amount of raw data in there, but we're also going to have some interpreted data sets which are a bit more explanatory. Actually, we are sort of thinking of branching that part of the project towards education as well, sort of to build it into a way that people can use to learn how tumors are made. So yeah, there are a few ways where if people is interested can get some more information and so on.
Just contact the CRUK Cambridge Institute or the Cambridge
they can contact the CRUK Cambridge Institute. We have a website on the Cancer Grand Challenge website. There's a sub-page, and they have an office that's very active in them passing information back to us so that they can contact them. I mean, somebody's interested, they can contact me, and I'll try to answer as well as I can. We are very open to interacting, but outreach and interaction with the communities is a big part of this. It was actually mandated by the project, right? CRUK told us they wanted us to do this, but it's also something that a lot of the people that's involved really genuinely like doing. So we have done a TEDx at some point We have been at the Cambridge Science Week. So whenever there is some event where there's this kind of outreach and connection opportunities, we try to be there as much as we can.
So you've talked a bit about the future of this project with the box. How do you see that coming to fruition? How do you see the project continuing?
We are working quite closely with CRUK to see if there's a way of keeping a legacy going on. So obviously we've got all this infrastructure, all this system that we hope will continue to some extent to exist even after the end of the Grand Challenge, even as a sort of service to other research projects. We're sort of thinking about what's the best way to make that happen. As I said, we've got a number of research projects, some of which still very basic research, some of which are a bit more applied, that are branched off of this, and they're going to keep going for 3 years, 4 years, something like that. So it's a gift that keeps on giving. So we're going to continue going there. And a lot of it is going to then sort of get closer to the sort of medicine aspect of it. It doesn't hurt that the Cambridge Research Hospital is being planned now. And we have very good contacts with the people that are behind it. I mean, the opposite, Richard Gilbertson, which is sort of chairing that, is our colleague, and we're collaborating together. The person that's been selected as the lead nurse of the hospital is our patient advocate of the Grand Challenge project. And we're thinking about a way of introducing the virtual reality there as a sort of permanent exposition that the patients themselves can actually use. So we are looking basically on all the possible ways that we have to keep this alive. And then in terms of technology development, as I was saying, we're starting to look at the next generation of technologies. We have got what's called a Pioneer Grant, which is a grant for sort of high-risk, high-gain development projects. And we are using it to develop a bunch of new technologies that ideally should sort of take us to the next step. The future, at least the next 3-4 years, are extremely busy, I think, on all aspects.
Yep. Sounds like you're going to have your hands full. Are you using virtual reality in any other research projects, or are there any plans to do so in the future?
So currently we're actually trying to gauge exactly that question. I mean, we have run a pretty big demo here in the Institute in the last couple of months, basically showing virtual reality to everybody that wanted to see it. And that was quite interesting because it's a tool that, although it was built for our type of data, it can be adapted very easily to other type of data. It can be used for any type of microscopy. It can be used for regular single-cell sequencing, which the Institute does a lot. Even can be used for some more basic technologies like flow cytometry and so on. Although, I mean, I said basic, and probably the head of flow cytometry is going to beat me up for that because they're really getting way more advanced than they used to be now. So, I mean, we actually have a lot of interest in adapting the virtual reality to answer slightly different questions. Actually, one thing that I was talking about was to use it to analyze multidimensional COVID datasets, where we were basically looking at the immune response of patients to SARS-CoV-2. And of course, the company, which we are trying to get funded now, will then offer this to any research group that is interested and support it. So I think that there's a lot of potential, there's a lot of interest. Currently, that's constrained by the limits of human body and the human mind, meaning there's like 3 or 4 people doing it and we cannot do everything. That's actually being a perpetual blessing and curse of the project. Like, we have always had a lot more ideas and a lot more collaboration avenues that we had resources to pursue. It's not just a matter of money, it's a matter of space, it's a matter of people, it's a matter of just energy. So we had to take the hard decision at some point to say, okay, this has to go on the back burner, otherwise we're going to lose focus, we're not going to get to the end line. So a lot of those things I hope that now we're going to manage to re-expand onto, and virtual reality for other research types is one of them.
How does this fit in with your broader research?
I can answer this both for Greg's laboratory and for myself. So for Greg's laboratory, cancer research is about maybe a half or a third of what the laboratory does. It has always been a laboratory with a lot of experience in cancer research, going back to when Greg was actually one of the discoverers of one of the main oncogenes. The other part of it, there's a pretty significant group doing small RNA biology and generally basic RNA biology. And there's another pretty large group of people that's sort of doing technology development, trying to inventing new methods. But the technology development really sort of fits back into the cancer as well, because a lot of these technologies then are applied to these other two fields. So I would say probably it's about half of what they're doing. For my own focus, I've been doing technology development and specifically in the field of spatial profiling mapping since I have started my career essentially. So right now that's about 100% of what I do. But of course, it's a very broad 100%. And going forward, I'm actually very interested both in looking at cancer, but also in using these technologies to understand the physiological state of organisms. There's always this flip side that you can study cancer all you want, but you need to understand how the normal tissue works in order to really then understand how the cancer development works. So I think that what I'm interested in looking ahead is to continue working on cancer, but then also using these technologies to look at how tissues work in their normal state.
My last question, I guess, is where do you see integrated cancer medicine taking us in the next 5 to 10 years? What's your take on that?
5 to 10 years. Okay, that's quite a bit of room to play with. I think in 5 years, a lot of the technologies that right now are research-based only, I think we're going to see them bleeding into practice, bleeding into therapy. Some of the technologies we are working with will do so. I think imaging mass cytometry will. Quite a few of the other things we are doing certainly will. And I would expect that some of these newer things that we're starting to look at eventually will as well. So definitely there will be, I think, an approach to cancer therapy that is a bit more customized for the patient. I think we're going to start seeing the fruits of that finally in the next 5 years. In the next 10 years, I think that's, it's kind of carrying that even further. I think that it's... we're going to be in a situation in which we're going to be able, hopefully, to diagnose a lot of cancers faster and to have a much better understanding on what happens in them after they've been diagnosed. And then I think that perhaps the thing that could have the biggest impact, that's not a prediction, it's a hope. The two things that I believe are going to have the biggest impact are diagnosis, because the fastest solution to any cancer is always yanking it out when it's still small. The other thing is understanding really the metastatic process, because ultimately, for the majority of cancer, what's really causing mortality, morbidity is the metastatic process. And for something that's so important, we still understand very, very, very little of it. Part of it is actually because a lot of the metastatic process happens when we don't see it. We see metastases when they're big. By that time, it's kind of late. It's not only late to treatment, it's also late to understand how they started, because at that point, the tissue is already all messed up. So I think that some of the technologies that we are working on now will allow scientists and doctors to look at metastases much earlier in time when they're just starting. And that in turn will allow them to get a much better understanding of what you have to do when you treat the primary tumor to try to minimize the risk of metastasis happening. And then I think that that's going to have a pretty big impact. I think that 10 years is probably a good time frame for them, 5 to 10, because there's understanding that needs to happen first, and then there has to be bleeding out into therapy. So it's probably a little bit longer than just taking the technologies that we have now and just putting them into therapy directly. But I think that that's what's really going to have a lot of impact on people.
Thank you so much. It just remains for me to say thank you for recording this really interesting conversation with me this afternoon.
Thanks to you. That was great to have a chance to chat and to talk about things.
If you want to find out more about the work of the Mark Foundation Institute for Integrated Cancer Medicine, please visit our website at www.integratedcancermedicine.org. where you can find details of the ICM vision, all the current research, clinical trials, resources, publications, and team information. You can keep up to date with our latest news and events, and you can also sign up for our newsletter. If you would like more information about the work of the CRUK Cambridge Centre, please go to www.crukcambridgecentre.org.uk, or you can connect with us on Twitter using our handle @CRUKCamCentre. Thanks for listening, and do join us again soon.